Good afternoon, ladies and gentlemen. Thank you, Dr. Jasuja, for inviting me here. I'll be speaking on a very small topic, ROS1 positive lung cancer and how to select. Actually, the selection part is rather easy for us because the number of molecules available is very small and only one of them is approved. So selection is not going to be a big deal. ROS1, a known oncogene, not only in lung cancer, but it is found to be rearranged in 22 different malignancies. In non-small cell lung cancer, only 1 to 2% of patients have ROS1 rearrangement, and typically seen in young patients, non-smokers or light smokers, and adenocarcinoma less common in other histological types. It causes ROS1 rearrangement causes fusion of a portion of ROS1 to its tyrosine kinase domain, and then brings about it remains constitutively activated and functions as a potent oncogene. It is phylogenetically related to ALK, and that is why there is a overlap between in the ALK positive lung cancers and ROS1, and that is why the drugs which we use for ROS1 are also the ones which are effective in ALK positive lung cancer: crizotinib, entrectinib, and lorlatinib. Kinase domain of ALK and ROS, again, they share 77% of amino acid identity, and that is why crizotinib also has shown a good binding activity to both ALK and ROS. That is why it was the logical first choice for lung cancer. Since it's seen in only 1 to 2% of lung cancers, we cannot have large studies for ROS positive lung cancers. So that is why this data is from other studies, and it has the ROS positive subset has been studied. So this is from the Profile 1001 study. It was for ALK ROS both, but the ROS cohort was only 53 patients, locally advanced patients, good performance status. Treated brain mets were allowed. They were treated with crizotinib 250 milligram twice a day, and the endpoint was objective response, duration of response, time to tumor regression, and progression-free survival. A fair mix of patients, Caucasians as well as the Asian population, and as expected, 75% of these patients were non-smokers. The waterfall plot is interesting. Majority of these patients have had responses. The overall response rate is 71%. We've also had 10% of patients who had a complete response, a rarity in patients who have an advanced lung cancer. And the duration of response is also good, 24 months, that's 2 years of response. So that is very impressive. The side effect profile, relatively tolerable. We've been using crizotinib for ALK positive lung cancer, so we know about the drug. The list is rather long, but the grade 3, grade 4 side effects are very, very small. If you can see it on the right-hand side of the slide. And overall survival was close to 51.4 months, close to a little over 4 years. That is impressive. So as number of, that tail effect is also seen here. The 1-year OS rate was 79%, but the 4-year OS rate was 51%. So at least half the patients were alive at 4 years. That is truly impressive. So based on this, crizotinib naturally got approved for frontline use in this population, but 1/3 of patients with ROS positive lung cancer have CNS disease either at baseline or during progression, and crizotinib does not have a good activity in CNS. In fact, it is sometimes the only site of progression in ROS positive lung cancers, and that is why we needed a drug with the better CNS activity. Entrectinib is a multi-kinase inhibitor, ROS, NTRK, ALK. It has a significant CNS activity, and it's more potent ROS inhibitor than crizotinib, at least in the preclinical studies. It has a good penetration in the brain and remains within the CNS. So that is why it was again a natural choice to use entrectinib. Again, small population, so this is not a study only for ROS positive lung cancer. It's a basket trial, and of which the ROS positive subgroup was analyzed separately. And on independent review, people who had the overall response rate was 77%. Again, good overall response. People with a CNS disease had 73%. Without CNS disease, even better, 80% of patients responded. So that is really good. The 12-month probability of PFS was 65%, and 12-month probability of overall survival was 85%. So that is really something. By and large, tolerable drug, we don't have any experience with this drug, but looks tolerable. The side effects, though long, the grade 3, grade 4 side effects are very minimal, and only 3.9% of patients had to discontinue treatment because of treatment-related side effects, but 1/4 of patients did undergo dose reductions, and serious adverse events were seen in 8.5% of patients. So we don't have entrectinib. We have another drug. The most common cause of crizotinib resistance is found to be a mutation in ROS1 G2032R, and that is shown to sterically impede drug binding. And this is going to be a hindrance as we start using more and more crizotinib. Lorlatinib is an oral Al-cross TKI that was studied in phase 1-2 trials in patients who were both naive as well as pre-treated with crizotinib, also in a cohort of ROS positive lung cancers. But as of now, lorlatinib is not approved for use in lung cancer. But nevertheless, the data looks impressive. 69 patients treated with lorlatinib, doses going from 10 milligram to 100 milligram, and overall response rate again looked fairly impressive. In TKI naive patients, the overall response rate was 62%, and patients who were pre-treated with crizotinib, the overall response rate was about 35%. The important thing is the time to tumor response. That was very small. So within 1.5 or 2 months, the patients did respond, and duration of response, especially in patients who were TKI naive, the duration of response is again more than 2 years, 25.3 months. And even in patients who have received prior crizotinib, the duration of response is over a year. So that is impressive. The waterfall plot also looks good. Very few patients who had progressed. Most of them had some kind of either complete or partial response, or some of them even had stable disease. So this is true for both intracranial as well as extracranial response. So ROS positive lung cancer selection, I was told to talk about selection. We don't have much to select. It's only crizotinib today, and it remains the standard of care. But intracranial and extracranial failures are going to be inevitable, and we need to have strategies to tackle these crizotinib failures. Probably entrectinib for better efficacy and lorlatinib for better intracranial response. They look promising. Thank you.